Hi, video 3 in the series of biochemistry videos. I want to deal with a couple of issues left over from the last video, and then today I want to talk about the structure of proteins. So last video, if I can just bring you back. I put this on the board. Hope you can see it. When I had a look at what I'd recorded, there's a delta plus there and a delta plus there. 2 delta pluses. Now that is a mistake. The carbonyl bond is polar. Delta plus on one side should be delta minus on the other side. So to make that clear, that there should be delta minus. If I talk about it with you, this kind of carbonyl, the carbon and the oxygen double bond, that carbonyl is polar, such that that side, the oxygen side, is delta minus and the carbon is delta plus. So the polarity should be that way around. Delta minus, delta plus. I apologize for my mistake. I don't want you guys making it. All right? We will make mistakes, but I want you to try and minimize the mistakes, and certainly I don't want you picking up my mistakes and carrying them forward. That's not the point. So carrying on with the lesson, what I really want to talk about and get onto is structural proteins. But before I do that, let's go on to talk about hydrolysis as a reverse reaction of the reaction we talked about recently, which is the joining of amino acids. So the joining of amino acids is condensation. 2 amino acids join together, they kick out water. That joining, eliminating a small molecule is known as condensation. So condensation reaction eliminates water. Now, that is a reversible reaction. I want to talk about that first. The water, if you had 2 amino acids joined together, the water will break them up. And reaction with water we call hydrolysis. So I want to talk about reacting water with joined up amino acids, and do we separate them back into individual amino acids? And yes, we do, but let's go through it in more detail. So to start the lesson, I talked about this last time, and I want you to know that the reverse of this, the equilibria, going backwards, the reverse of this is hydrolysis. And the hydrolysis, I want to just take you through because it's an important reaction. And the way I want to do it is I want to consider, say you've got an amino acid 1 and amino acid 2. So 2 amino acids, bring them together. You guys know that this will react to give you so this circle, the circle represents the rest of the molecule, the square represents the rest of the molecule for amino acid 2. And when they come together, regardless of which amino acids it is, they will come together with the amine of one amino acid reacting with a carboxylic acid of the other. And consequently that makes that peptide link. So the peptide link will link amino acid 1 with amino acid 2. And we can carry that forward. We can have more reaction on this side, peptide link, the next amino acid attaches on this side. Equally, the carboxylic acid can react with the amino acid of another amino acid, sorry, the amine group will react with the amine group of another amino acid, and then you get another attached amino acid on that side. So the number of amino acids linking together can grow from 2 to 3 to 4 to 5 to 6 to many. And essentially that's what I want to talk about later, is the structure, the primary structure of proteins. However, let's discuss, will water take this reaction backwards? Will it be a reversible reaction? And will the water, and how is it going to break that dipeptide? How is water going to break that dipeptide down in a reaction called hydrolysis? Well, if you understand that water, oxygen, has a lone pair of electrons, there, and then if you understand that that lone pair of electrons can go and attack the electron deficient carbon, let me draw that the correct way around, see? Delta plus, delta minus, the pair of electrons, and there's more than one pair of electrons on the oxygen. There are in fact two lone pairs of electrons on that oxygen. But I'm just going to use one of them. So as you can see, there will be an attraction. There will be an attraction where the lone pair of electrons makes a dative covalent bond with that carbon. As it does so, that double bond will open. We're familiar with that. The double bond opens, and then the double bond closes, and then that bond, the carbon-nitrogen bond, pair of electrons, will move onto the nitrogen. And that causes a break. Now, when it breaks, what do we get? Well, let me draw you what we get. We're going to get on this side H2N with that rest of the molecule represented by a circle, joined to carbon, doubly bonded oxygen has been reformed back again, but now we've got oxygen, we've got hydrogen, hydrogen, and on this side we've got nitrogen, hydrogen, square with a COOH. Now, because this is neutral and that pair of electrons goes in there and that pair of electrons is then being shared, the oxygen has overall lost its share of one electron and therefore the oxygen is positively charged. Same kind of argument here, the two electrons moving on to the nitrogen. Now the nitrogen has that pair of electrons on its own, so it's kind of gained an electron in its taking the two electrons for itself, so there's a negative charge there. Well, what will happen as a consequence next is that this side will lose the proton, so this will lose a proton like that, and the H plus, this is a H plus here, the H plus, that breaks off if you got H plus, and that pair of electrons there will pick up. That H plus is that H breaks off as H plus, that H plus onto there regenerates for you the two amino acids you started with. So you end up with H2N bond circle 1COOH, and you end up with NH2 square COOH. Can you see that the hydrolysis reaction has taken us back to the amino acid 1 and amino acid 2 that we started with? So, I'll put that back in a minute, but I want you to know that this joining together amino acids is reversible, and water will cause that to go backwards in a hydrolysis reaction. The problem with using water to break these peptide links and regenerate the amino acid that we started with, the problem is the reaction with water is a bit slow. So we can catalyze, we can use a catalyst to do this reaction, and the catalyst that works better is hydroxide ions like alkaline hydrolysis. Or you can use acid hydrolysis. You can use acid as the catalyst. But let's just think why acid or alkali. You should be able to get your mind around the alkali argument really quite easily, so I'll go through that. Will work faster than water. So here, this OH, the oxygen on the water. Water is not such a good base. Hydroxide ions are a much better base. Think, what if I had HO instead? If I had HO instead, water is neutral, is attracted to the electron deficient carbon. This is negatively charged. Hydroxide ions doing that opening, that closing, that breaking will happen much more readily. The hydroxide ion will be a better base. The hydroxide ion will react faster as a consequence of it being attracted stronger because it's got a negative charge on it, and this breaking, this cleavage of the peptide link will happen much more readily. The thing that you've got to be careful about is that if you use hydroxide ions, then here under alkaline conditions, in alkaline conditions, then that and that won't be there. These will make the carboxylate ion end to the amino acids that we've got. So what changes with using alkali is it's entirely the same reaction. It's like the reaction with water. It will break the peptide link, it'll regenerate the two amino acids we're talking about. However, you've got to be mindful of one of my previous videos where I said look at the conditions. If the conditions are alkali, then the alkali will cause your carboxylic acid group to lose its proton. And similarly, if in an exam question they wanted you to do acid hydrolysis and use an acid to break up the link, the peptide link, the same thing would happen as I've been through with water, except at the end the acid conditions would protonate the amine groups. The amine groups would be protonated. So bear in mind what conditions, but the reaction is just the same. The reaction is the reaction I've just been through with water. If you need to write an equation, then the reaction is with water, and the alkali or the acid is just a catalyst that you don't need to include in the equation. Okay, so if we now move on to talking about the structure of if we now